Okay, at the count of three. 1, 2, 3. Hit enter. Let's see your answers. Okay, good. Okay, awesome. I see some good answers. I did notice that we're still practicing, right? So remember, RNA has no teeth. So if we're putting down T's, we're doing that right if it were DNA. If it were DNA, we would put T's, but in RNA, we don't. And it's going to take us a little bit of practice. So the original sequence was TTC, GCT, ACT. So we should have put, you can check your work, and everybody's going to get points regardless. AAG, CGA, UGU. No, UEA. So the reason we put an A here is the reason we put a U here is there's an A here. The last sequence is ACT, ACT. A goes with U, C goes with G, and T goes with A. So everybody got some points here for participating. But if you put a T, if it were DNA, you would have been 100% correct. And this is where most students are like, wait, what's going on? Like, why? So just remember, okay? This is important. We're going to use this in a sec. And the state of Texas will try to, kind of bank on that. They're going to forget how RNA works. A will always go with U in RNA, and C will go with G in RNA. In DNA, if we were copying DNA, A goes with T, and C goes with G. So you just have to read the problem carefully and decide, are we going to make a copy of DNA, and we'll go A to G, or are we going to make a messenger? We're going to make a messenger that we're going to use to use. Because RNA does not understand, like it doesn't have any teeth. It can't put something that it doesn't own. So let me go ahead and we'll come back here and you guys will practice in your notes one more time. And then we'll move on to step 2. So we're talking about pregnancy and anatomy. Those are some things there. Okay. So go ahead and in your notes, find this sequence right here. See where it says DNA template. It's underneath the picture with the green enzyme. Here's what we're going to do to make it a little bit easier to read. In reality, when the messenger or when the message gets read here in a little bit, because right now we're doing is copying it, it's going to be read 3 bases at a time. What that means is it's going to read it 3 codes at a time like this. So just to make it easier for us to read, so it doesn't look all jumbled up together, I encourage y'all to break it up into 3s because you're going to see that that is how the sequences are read. So right now when you work on your problem, you're going to see that they're spaced out in 3s. Just like if you read a book, you kind of start from the left to the right, 1 page at a time, 1 word at a time. The messenger, once it's read in the second step, it's going to be read in a very specific way. So all I did is I took my sequence here that we have and just spaced it out. Okay, this is in your notes, and you'll submit this probably on, I don't know if we'll have time today. We'll see because we have a test, not a test, but it's a learning checkpoint on Wednesday. Go ahead and before we move on to step 2, let's practice that again. Let's practice transcription. So again, let's say that this is a sequence for how to make melanin. The messenger needs to go in there and copy that, or this is a sequence on the virus on how to make spike protein. The messenger is just going to copy that. So you have an A, you're going to put a U. You have a T, you're going to put an A. You have a C, you're going to put a G. So go ahead and type it. I don't want to do it for you because y'all need to practice, and then you're going to check your work with mine, and then if you need to make any corrections, you're going to go ahead and do so. I'm going to stop sharing my screen for a sec so I can type it out, and then you can check your work. All you're doing is you're finding the partners for those sequences. Okay, so check your work with mine. So this is what the DNA template had on how to make melanin, which is our skin tone. A messenger would copy it as follows, or transcribe it as follows. It would put, since there's an A, it would complement it with a U. If DNA has a T, it would have complemented it with an A. D with G, G with C, C with G, G, PT, that goes with AA. A would go with T in DNA, but because we're in RNA, it goes down to U. GC, we have CG, and then T can still go with A because RNA still has an A. I'm going to put here no U's. You should never, I'm sorry, no T's. You should never put down a T in any of these problems that are asking you to find the mRNA strand. You gotta care about them. I tried to line it up so you can see where it came from, but if I can do that like that. That's what your sequence should be. This is all the step one is in making a protein. This is called transcription. All that the messenger did is it went ahead and copied or found the complement, the partner for the DNA strand. That's the first step to making a protein, and we call this transcription. The RNA, the messenger RNA copies the DNA. That's what's happening in this picture. See how the DNA unzips? And then the RNA, it lays down the complement. T, it put an A, it put a U, C, it put a G. That's all it's doing. I went to my mom's house, hey mom, what's the recipe for how to make a chicken noodle soup? Okay. This is called transcription. But we don't have a protein yet, so we're going to continue. We're going to meet our next set of workers. In order for me to be able to make the chicken noodle soup, I need to read what I wrote down, and then I have to go get the ingredients at HEB, and then I have to cook it. So in the next set, we're going to see that now we need something in the cell to read the instructions, to read the message. Otherwise, what's the point of taking a message if no one's going to read it? So we're going to have a reader, and then we're going to have the ingredient guide come and bring all the ingredients needed to make that protein. So the next key player that we're going to meet here in making a protein is what we call rRNA. So rRNA stands for ribosome. The ribosome now is going to make the protein from the instructions that the messenger brings. And we're going to see what this looks like here in a moment. So rRNA stands for ribosome, ribosomal RNA. I also think of this one as the reader. This is the one that's going to read the message now and then make the protein. So it's also kind of like the cook because it's going to physically make the protein. All cells have ribosomes. And let me show you what a ribosome looks like, just in case we need to kind of review. I know it's been a while since we saw one. Ribosomes look like little tiny specks or dots inside of the cell. That's all it is. These little dots inside of the cell are ribosomes. This cell has a nucleus, therefore it's a eukaryotic cell. Those are ribosomes. The ribosomes can be floating around in the cytoplasm. They're the protein factories, or they can also be on the rough ER. So it's got a bunch of little dots on there. Actually, this picture is probably larger for you guys. So those are ribosomes, and that is what the rRNA is. These are our little protein factories. All cells have ribosomes because all cells make protein, so bacteria have them too. Okay, so let's go ahead and define our RNA as the reader, or what it's going to do its function. What does it look like? So I showed you all a picture. Let me show you another picture. Plants have them, all cells have them. But I want you to describe what they look like. So one of the things they all are going to be responsible for is recognizing what they look like. So here the ribosomes is little dots. Here they're attached. So in your own words, what do ribosomes look like? I would probably just type in and look like little circles or look like little dots. What do they look like to you? Let's define what ribosomes do with the rRNA. What the rRNA is going to do now, it's going to read the message that was copied from the DNA. It's going to read the messenger. And it's going to read the messenger 3 letters, what we call basic codes, at a time. The ribosome is also going to make the protein. So going back to that story I was telling you, so my mom would have been like the DNA. She had the instructions, the recipe on how to make the chicken noodle soup. I wanted to make the chicken noodle soup, so I went to my mom's house and I copied down the recipe from what she was telling me. But see, I'm not gonna make the soup because let's say my kitchen's under construction. I'm having my house remodeled. So I go over to my sister's house and I tell her, hey sis, I got this recipe from mom. Do you think you could make it for us? And then my sister would read it. Okay, we're gonna need chicken broth, we're gonna need noodles, we're gonna need carrots, and then we're gonna use her kitchen and she's gonna cook it. Okay? I just brought her like the recipe, even though I'm the one that wants to make it, she's the one that's gonna make it for me. Okay? That's what the ribosome does. It's gonna read the message 3 bases at a time. So this process here is now called translation because the ribosome is going to take the message that was copied from the DNA and translate it into a protein. It's going to read it, figure out what it's going to need to make that protein. How are we going to make more melanin for this person that's been out in the sun all week? So what we can see here is a little bit of the key players. For starters, we see the messenger RNA. We see like a single strand. And then we see the bases, A, U, G, A, so we know it has to be RNA. The other thing that we see is kind of see this blob right here. This is the ribosome. And what it's going to do is it's kind of reading it. It's decoding it. We also see these little guys. They look like paper clips. We haven't gotten there yet. But this process here is called translation, and it's actually going to make the protein. Another image here is, here's the messenger that was just copied by the DNA that you guys just practiced doing the transcription. C, G, U, so it got this complementary sequence from the DNA. And now the ribosome or the rRNA is going to read it 3 bases at a time. So in order to understand what CGU means and GAC means, we're going to use a codon chart. So we're going to learn how to read that. So let's take a look at what we're looking at here, because this can be quite intimidating, but we're going to practice how to use it. So now you are the ribosome. You just got this message on how to make a specific protein. Let's say you just got vaccinated for COVID. Your cells just got a message on how to make COVID spike protein. And you're going to read it and figure out how to make those proteins. And so if you guys noticed here, and I'm going to take this and I'm going to copy it because I want to write on it. So I can show you how to interpret this thing. And this skill that we're about to learn, you're going to use to make a little alien this week. You guys are going to read the genetic sequence of a fictional alien. And you're going to go through this process. So it's really important that you learn how to work through these. I'm trying to find words. Right here. Okay. I'm going to zoom in a little. A lot, actually. You guys have this. I'm just doing this here because I want to be able to write on it. I want to show you a couple things. You guys are going to notice the sequence of letters or bases here. See that? It has U, C, A, G. Those are the nitrogen bases. That means I have the U. And it says first letter. Then at the top, you're going to see another sequence of 4 letters or bases. The second letter. That sounds like. So those are second base. And then you're going to see on the right-hand side a sequence of letters again. Now we see them repeatedly, U, C, A, G. U, C, A, G. You see them again. It's just kind of repeating. And this says third letter. Before I continue, can you all just confirm you can see my screen? We're good, you can see it? Awesome, thank you. All right, so the way that you're going to use this codon chart, well, first let me kind of explain what's going on. You guys are going to see on the inside of this codon chart these very odd words. Asparagine, lysine, arginine. Those are amino acids because in order to make a protein, the building block of a protein is an amino acid. And the cool thing about these guys is all life forms have the same 20. There are actually 20. If you were to count how many unique, like non-repeats there are 20 amino acids. Plants use the same 20, animals use the same 20, mushrooms use the same 20, bacteria use the same 20, which is great because that means that you don't have to eat another human being to make human proteins. You can basically eat any life form and get the ingredients that you need or the amino acids to make a protein. So we recycle the amino acids from the living things that we eat and then we put them in the correct order. Viruses obviously use the same amino acids because you're using these to make the spike protein on the COVID virus. So once the ribosome gets the message, it has to decode it. And this is the decoding part known as translation. And the way that you're going to translate a genetic sequence is the message is going to be read 3 bases at a time. So let's say that the message came with the sequence AUG, right? That is a sequence that the message got from the DNA. The way that we're going to read this is we're going to read it, so there are 3. We're going to basically call the first letter 1, the second letter 2, the third letter 3, and these are known as nitrogen bases. So you guys see how on this codon chart it says 1, 2, 3. If the first base is A, we're going to go ahead and start here with this first letter, and we're going to find where the A is at. If this is the A, you guys see how they're broken up into like quadrants? Like this is a quadrant right here. This is a quadrant right here. This is a quadrant right here. And then this is another little section right there. Since the first letter is A, we're going to focus only on this section right here. Our answer is going to be somewhere in this section. We're going to ignore the rest for now. We're just going to use this section. So our answer for the first sequence is going to be one of these nitrogen bases. Change my color just so we'll call this in. All right. The next letter, or this next base, is U. There's the R at the top. It says second letter. I'm going to find U. And I could be here or here or here or here. So U could be anywhere here in this section. But remember that we're only going to work on this quadrant right here, this little section, because we're focusing on the A region. So our answer is going to be somewhere here. Why? Because it's AU. So A and U, like if you guys, you know how you graph in math, the like X and Y axis? Kind of what's happening here, A and U intersect somewhere here. And the ribosome is doing all this. The last sequence is G. So you guys see where it says third letter? There are a lot of G's that I could choose from. I could use this G, I could use this G, I could use this G, or I could use this G. But because we're focusing only on this little region now, I'm going to use this G, and I'm going to go all the way across until I end up in this section. And the amino acid, the ingredient for how to make that protein, the first ingredient is methionine. That's what AUG code is for. But instead of writing out the whole word, what you see a lot in these problems is methionine. They're just going to take the first 3 letters and just write this down. That is the first amino acid or ingredient on how to make a protein. So in a moment, when I pass this out, in this particular question, they wrote it all down. Like they wrote out the whole amino acid. Sometimes they'll just give you arch or met or loop. That's pretty typical as well. So if you see like an amino acid, like this in an answer choice, it just says ALA. Based on your choices, ALA would have to be this one, alanine. So sometimes you'll abbreviate it just with the first 3 letters, and sometimes you can get the whole thing down. This is the amino acid that ribosome is going to need to make the protein. This process here is known as translation because it took this sequence, AUG, which came from the DNA, to make a protein here in a moment. Okay, so let's go ahead and take a look at that in our notes. And let's practice. I have the exit ticket here, but I don't know if we'll be ready to work on it right now. All right, I'm looking for the notes. Okay. So what you're seeing here is here's a messenger. Here's what the ribosome is doing. It's the reader. It's going through all this work. You guys see how you already have the sequences AUG, or sorry, UAG, CGG, AAU. What you're going to do now is take those, use your codon chart, and figure out what those sequences mean. So let's do one together, and then I will, you're going to be your DCA instead of because I have a question set up for you guys, but I'm going to actually do this as like a first question next time, not on Wednesday, because you all probably will see these questions on Wednesday. And so let me just talk about the test a little bit because we're going to get ready here to go in the next like 12 minutes. When we meet on Wednesday, we're going to take a learning checkpoint because we do have, the state is still has the expectation that all freshmen have to take their STAR test in order to fulfill their graduation requirements. Whether you choose to do that in May or in June or as a sophomore, that's also going to be ultimately your choice, but you're still going to be asked to take this test to graduate. And so, because we're in biology now, we're going to go ahead and focus on this as if we're taking the test in May, which some of you did share that you will be coming to campus. If you're like coming to campus already, the expectation is you're going to be here to take the test. But if you're remote, you can choose to just come and take the test, and then that's it. But in May, it's an in-person thing. Anyway, so Wednesday, we're going to be taking the test to see where we're at. How are we doing? How are you doing with the concept? So you have an idea of where you're at a couple weeks before, and we can start focusing on the things that we need to brush up on. And so there's a good chance that you're going to see some of these questions, even though we're not there yet, just to kind of see where we're at. It's a checkpoint. So I don't want to ask you this one just yet because I want to see, first of all, if you all are understanding how to read the codon chart, because I'll tell you right now, like most students, at first, it takes quite a bit of practice. So you guys are going to be working on an alien to practice. So for our DCA today, we're going to submit this, even though it's not done, because we haven't finished the entire box. But I want you to submit this before you go today, because I want to see how you transcribe this sequence. So I'll do the first one with you, and then on your own today, before you head out, you're going to do the rest couple of sequences, and you're going to submit it. And I'm just going to check your amino acid sequence to see where we're at. So let's do the first one together. The first sequence is UAG. What we're doing is translating. So now we're going to read the messenger as if we were the ribosome. We're going to use our codon chart. So the first letter is U. So we're going to go start here. Our answer is going to be somewhere in this section. The next letter is A. That's the second letter. So U and A intersect in this quadrant. Let's see that. UA. And then the third letter is G. So there are a lot of G's to choose from, but we're going to use this G because we're going to intersect right here. U, and you can't see my hands, but U, A, and G codes for stop. Now, what does that even mean? There is actually a protein that tells our ribosome stop. Stop here. This is where the protein is. So this protein is, let's say, a mutation, and it stops before it even begins. But just we'll ignore that, but this codes for stop. We're just practicing right now. Normally, stop would go at the end, like the period at the end of a sentence. This is how to make a COVID protein, or you stop here. It's all the instructions. So UAG, why did that code for stop? Because the first letter was A, so the answer will be somewhere here. I'm getting thrown off of my head. UAG, U is here, A is here, and G is here. Codes for stop. Okay, so before you head out, this is what your DCA will be for today. Go ahead and translate. You don't have to highlight them, but translate the next couple of sequences to see what amino acid they code for. And when you do, continue on the sequence. And then you'll submit. I'll post my codon chart so you can see a little bit better. I did record the session so that if you need to go back and pause, you can do that. I'm going to go read the chat box real quick. I'll do that there so you can read the sequence and make process. it? Yeah, once you submit it, okay, so what you're gonna do is you're gonna submit this, just hit submit. Obviously, it's not all done, but I just want to see how you all transcribe this sequence. And then almost done for you. I'm gonna use this as your check-in for you guys. Yes, I'll scroll up, Cindy. I just saw it. I'm gonna read the chat box. See if any have a question. That is Alright guys, it's 9:03. You're gonna turn that in. You should just transcribe, translate it, as highlighted portions of the messenger. Just submit on your Google Classroom. Lauren, are you still here, Lauren? Once you submit that, y'all can end the call. It's been at C17, you're still here. I'm gonna end it at 9:05. Okay, bye guys. Have a great day.